Omicron-neutralizing antibodies found. Scientists from across the world have discovered antibodies that are capable of neutralizing the Omicron virus and other SARS-CoV-2 subtypes. Antibodies to the viral spike protein are directed towards parts of the protein that have remained relatively constant throughout the evolution of the virus. Broadly neutralizing antibodies on the spike protein could lead to vaccines and antibody treatments that are effective against not only Omicron variants but other variants that may emerge in the future, according to David Veesler, an investigator with the Howard Hughes Medical Institute and associate professor of biochemistry at the University of Washington in Seattle, who led the research. According to Veesler, this discovery shows us how to resist the virus' ongoing evolution by concentrating on antibodies that target these highly conserved regions on spike protein. A collaboration between Humab's Biomed SA's David Corti and Ver Biotechnologies. Veesler was the driving force behind the study. Nature published the study's results on December 23. Elisabetta Cameroni and Christian Saliba, Humab's, John E. Bowen, UW Biochemistry, and Laura Rosen, Humab's, were the study's primary authors. The spike protein, which the Omicron variety utilizes to attach onto and penetrate cells, contains 37 mutations in the mutated form. A large number of mutations have occurred in this sample. Because of these alterations, the variety has been able to spread so quickly, infect those who have been vaccinated, and reinfect those who have already been infected, according to some experts. There were a number of issues that we wanted to answer. How did these mutations in the spike protein, which are seen in this version, influence its capacity to connect to cells and elude the immune system's antibody responses? Veesler asked. According to Veesler and his colleagues, the enormous number of mutations in Omicron may have occurred as a result of the virus hopping from people to animals and back again, or as a result of a lengthy infection in someone with a weaker immune system. The researchers developed a pseudovirus, a crippled, non-replicating virus, to generate spike proteins on its surface, like coronaviruses do, in order to test the effects of these changes. Once they got the spike proteins with the Omicron alterations and those detected on the early pandemic variations, the next generated pseudoviruses that contained the same spike proteins. Scientists began by testing how effectively the various spike proteins bound to proteins on cells' surfaces, which the virus utilizes to grab onto and penetrate cells. The ACE2 receptor is the scientific name for this particular protein. Omicron spike protein was 2.4 times more effective at binding than the spike protein identified in a virus obtained at the beginning of this epidemic, according to researchers. When SARS broke out in 2002-2003, changes that enhanced affinity in the spike protein were linked to increased transmission and infectivity. Researchers discovered that Omicron could attach to the ACE2 receptors in mice, indicating that it may ping-pong between humans and other animals. Antibodies raised against previous strains of the virus were tested to see whether they could defend against the Omicron form as well. Their method used antibodies from individuals who had either previously been infected with the virus, had been vaccinated against earlier strains of the virus, or who were already sick with the virus but had been successfully treated with the vaccination. All 6 of the most widely used vaccination antibodies tested were less effective at blocking infection than antibodies from healthy individuals or those who were immune to older strains of the virus. A single dose of Johnson & Johnson didn't stop the Omicron variant from getting into cells from people who had been infected before, people who had the Sputnik V or Sinopharm vaccines, or people who had both. All 3 vaccines' antigens, Moderna, Pfizer, BioNTech, and AstraZeneca, were significantly less effective in neutralizing the virus after 2 doses than any of the other 3. 2 vaccination doses lowered antibody activity, although the decrease was less dramatic, at around 5-fold. This clearly shows that vaccination after infection is beneficial. Moderna and Pfizer, mRNA-BioNTech's vaccines demonstrated a 4-fold decrease in neutralizing activity in the antibodies of renal dialysis patients who received a 3rd dosage of the vaccinations. That a 3rd dosage is effective against Omicron, Veesler said. In the laboratory, all antibody therapies, with the exception of one, that are presently permitted or licensed for use with patients who have been exposed to the virus, showed no or significantly decreased activity against Omicron. An antibody termed sotrovimab was found to have neutralizing activity that was reduced by 2 or 3 times, according to the research. Antibodies developed against previous generations of the virus were evaluated, and the researchers discovered 4 distinct types that were still able to neutralize Omicron. There are 4 distinct regions of the spike protein that are targeted by different types of SARS-CoV-2 and a group of closely similar coronaviruses known as sarbecoviruses. As a result of their critical role in the protein structure, these sites may continue to exist. As a result, they are known as conserved. A wide range of new viral variations might be targeted with vaccines and antibody therapies that target conserved regions, according to Veesler, since antibodies can neutralize via identification of conserved sites in so many distinct virus variants.